Eek. Morning, everyone. Thank you for that incredibly kind introduction, Malara, and thank you to the organizers for their bravery in inviting me to speak here. I'm here representing my lab and our amazing network of collaborators, and I know I speak for the whole collective when I say what an incredibly kind of humbling honor it is to have the opportunity to present our work before you. It really means a great deal. So the theme I'm going to try and address today is the theme of integrative neuroimaging of the developing brain in health and disease. And I'm going to approach this theme from a starting point in medicine, but as you'll see, we're going to go kind of far into basic science and back again. Just for some context, this is where we are. My group, the Developmental Neurogenomics Unit, or the DNU, lives here on the scenic 4th floor of the NIH Clinical Research Center. It's a large research-dedicated hospital outside DC. My own background's in pediatrics and child psychiatry, and the incredible group of people shown here that I'm lucky enough to work with represent really diverse disciplines: mental health nursing, developmental psychology, computer science, molecular biology, and systems neuroscience. And together, with ourselves and collaborators, we're trying to, our clinical goal, although as you see we reach into basic science, is to try and better understand the biology of neurodevelopmental disorders in ways that might ultimately help to improve disease prediction, detection, and treatment. So what do I mean by neurodevelopmental disorders? Well, the conditions we're concerned with are currently called things like autism spectrum disorder, ADHD, tic disorder, intellectual disability, and specific learning disabilities, to name a few. These conditions are currently diagnosed behaviorally. They're diagnostic categories, but in reality, they really have very fuzzy boundaries between each other and with normality. They're collectively common, affecting 1 in 10 people. They onset early in childhood, all with a male bias, and they're often lifespan persistent, making them, a considerable public health concern. And notably, they're highly comorbid with each other within individuals, and they tend to be clustered in families, which is already a clue that there might be biological interdependency between these objectively, supposedly discrete diagnostic categories. Now, huge effort has been poured into trying to find biomarkers for neurodevelopmental disorders, and I'm tagging this through the proxy of number of publications per year. And no doubt, we've learned a huge amount through this work. Unfortunately, though, we still haven't found a single biological signature that reliably segregates with one of these diagnostic categories, or helps us stratify people within a category to usefully predict outcome. So why might this be? Well, sorry, I'm just trying to get this projector. There we are. So I think there are 3 key challenges that I'd like to discuss, and the good news is I think each of them has an opportunity hidden within it that face us when we try and engage in biological research on neurodevelopmental disorders. And what I want to do is go through these challenges and opportunities as they frame the data that I'm going to get into in the latter part of the talk. So the first challenge is the staggering complexity in the architecture of genetic risk for neurodevelopmental disorders. So I'm showing you the diagnostic categories here, and underneath I'm showing you a particular class of genetic risk that I'm interested in studying, copy number variations. So neurodevelopmental disorders are some of the most heritable conditions we know, but they have this very complex architecture to genetic risk. So for those of you not familiar, copy number variations are when you're missing or have too much of a particular chunk of your genome. And that chunk can be a whole chromosome, a whole Y chromosome, X, chromosome 21, Down syndrome, or you can have deletions or duplications of little bits of chromosomes, which are given these addresses, chromosome 7, Q11. And as you can see, having a dosage abnormality in any one of these regions simultaneously increases your risk for multiple diagnostic endpoints. Conversely, having any one of those diagnoses, you can get to it from multiple genetic starting points. So it's a really gnarly many-to-many mapping problem. And so immediately you can see, if you're trying to find a biomarker for autism spectrum disorder, you're immediately lumping together a group of different genetic conditions, if you like, as defined by their genotype basis. And you may even be assuming that some of those genetic variants are specific for autism when they might not be. So it's not all bad news, though. I think this actually offers us an immense opportunity. We can leverage this genetic heterogeneity if we flip our strategy on its head, if we try and understand the biology of neurodevelopmental disorders, starting with genetically defined groups. And I think of this as a kind of hadron collider experiment for the brain. So the Large Hadron Collider, the idea is you sort of smash particles together, see what happens, and in doing so, understand the substructure of the nucleus. And in a partly analogous way, we can think of these rare genetic disorders as sort of stresses that we hurl at the brain. And if we can document what the brain does in response to these diverse genetic insults, perhaps that helps us reconstruct pathways of risk to neurodevelopmental disorders. I want to highlight a really beautiful example of this, I think, from led in a paper by Jacob Elgood from the lab of a good friend and collaborator, Jason Lurch. And what they did is they took over 20 mouse models of high-impact risk genes for autism. They put them all through the same structural imaging pipeline, and they measured what each genetic variant did to different anatomical chunks of the brain. So then you get this genotype by phenotype matrix. And then they submitted this matrix to unsupervised clustering. And what they got back was sets of disorders that are similar to each other through the spatial pattern of anatomical change they induce in the brain. And then conversely, they got distributed sets of brain regions that are defined by the fact that they share their pattern of vulnerability and resilience to genetic insult. So in a very elegant way, helping to simplify the space of genetic risk and anatomical responses. Sorry about this, I'm trying to... So unfortunately, a second challenge facing researchers in neurodevelopmental disorders is kind of development itself. So what it takes to get to a mature organism from a single cell involves the staggering coordination of genetic and environmental factors over time to lead to an incredibly spatio-temporally complex organism. Now, this process is stereotyped, but within that stereotypy, there's inter-individual variation. So we all have eyes, nose, and a mouth, but we don't all look the same. And rather awkwardly, it's highly protracted in humans, making it very hard to model in alternative systems. And it makes normal a moving target. It's very difficult to say, what is the disease signature now? What is it now? What is it now? But I think really more profoundly, development isn't a canvas that disease effects are painted on. It's more like a construction out of clay, which actually development itself builds the disease effects and can itself be modified by disease. And I think it's this deep interdependency between disease and development which offers a profound opportunity, as kind of communicated by this Escher etching, which is that if we recognize that disease effects unfold within developmental patterns, then we can try and learn the language of normal development, and in doing so, be able to ask deeper questions of the atypically developing brain. So the third challenge I want to discuss is that of scales. The organ we're interested in is highly complex, plastic, and locked in a bony vault that we can't get inside during life. We have amazing tools which are celebrated in this incredible community, which allow us to probe the brain at, say, millimeter resolution, and that's enabled us to do phenomenal things. But as is clear, there's an awful lot going on inside a millimeter cubed. So if this is what is inside the poppy seed-sized brain of a fly, and if the biology of neurodevelopmental disorders ultimately is rooted in genetic, molecular, and cellular processes, we really need to try and squeeze more information out of the sort of spatial maps that we can get from neuroimaging. Well, I think there's a huge opportunity here, because we're blessed now with a growing number of cellular and molecular maps that have been registered to neuroimaging space. I'm giving you a couple of examples here. The big brain from McGill, a 3D reconstruction of histological sections at 20 micron resolution, Nissl stained, and you can derive MRI-registered maps of cortical layer thickness, as was shown in the beautiful talk this morning by Conrad Waxtel. Another resource that I'll be using a great deal during this talk is the Allen Institute for Brain Sciences, which provides spatial maps of gene expression for about 20,000 genes in the adult human brain. So the fact that we have these histological and molecular maps lets us now carry out spatial comparison tests. And I want to highlight here another tool that I'll be using throughout the talk. This is what we call the spin test, published and developed with Aaron Alexander Block. And the idea here is basically, if you have any two maps that are in standard imaging space on the cortex, you can project them onto a sphere, and you can calculate the observed spatial correlation between those two maps, say this red bar here. And then you can just spin one of the maps 1,000 times, stopping it, spinning, stopping it, and recalculate that correlation to give you a null distribution. And these sorts of approaches let you make safe inferences about how similar are these two maps, this map of anatomical change and perhaps this map of cytological features. This is an immense opportunity opened up to our community now. So, very much like John Snow better understood the spread of cholera by co-registering a map of cholera deaths with a map of water pumps, perhaps we can better understand the biology of neurodevelopmental disorders by co-registering some of the maps we get from in vivo imaging with some of these newer cytological and molecular maps. So my group strongly is infused by these ideas, and we pursue the approaches that I've just mentioned through three main integrative avenues of work. In one, we carry out this sort of hadron collider experiment by deep phenotyping genetically distinct disorders that increase risk for neurodevelopmental disorders. As we ask questions of the brain in these genetic disorders, we listen to the information we're getting from our studies of typical brain development, which delivers us more kind of biologically valid phenotypes for querying the patients. And when we look at the maps that we get from typical brain development from our patient groups, we try to decode them using the growing resource of cellular and molecular atlases. And I want to sort of dig into this with some hard data in the rest of the talk. And I hope you bear with me through a cheesy device I've chosen to use. It's going to be a data story in 3 acts. Like any play, it's got its lead cast and characters, the amazing mentors and funders that have inspired and made this work possible, Emily Simonov, Judy Rappaport, Jay Geed, the fabulous team of colleagues and collaborators that I'm lucky enough to work with, that I'll mention throughout the slides as the work is presented, and the amazing patients and families that give their time to make this research possible, the incredible public data sets like the Allen Atlas and the Human Connectome Project. And in the corner there is what I have a sort of secret love affair with, I guess, is the T1 weighted structural MRI scan, because I'm constantly amazed by how much it can tell us, and I hope to share that sense of amazement with you throughout the talk. Okay, so in Act 1, we'll see that patient neuroimaging prompts a deep question for basic neuroscience. And our starting point is trying to figure out changes of brain anatomy in patients with sex chromosome aneuploidy. So this is the Hadron Collider experiment part of our work. We focus on sex chromosome aneuploidies as models of genetic risk for psychopathology. So normal sex chromosome dosage is XX for females and XY for males. 1 in 400 people are born with an extra sex chromosome. The best known is Klinefelter's, males with an extra X, but you can see there's this whole kind of variation of adding X's and adding Y's and multiple numbers to males and females. And this is where what we try to do is we recruit people with aneuploidy, we deep phenotype them using the same protocol, and then try to empirically retrace the breadcrumb trail from the gene dose abnormality to the surface changes in cognition and behavior. And the beginning point for our story is this fact, and the motivating fact, that these sex chromosome aneuploidies, they increase risk for diverse neurodevelopmental disorders, but they really seem to target language functioning and social functioning. So our question was, well, are these pattern defects of different gene dosage abnormalities on cognition and behavior accompanied by spatially patterned effects of these aneuploidies on distributed systems within the brain? And we approached this using the T1 weighted structural imaging scan in this sample of about 300 people that represent diverse sex chromosome dosages. So just to orient you, our question was, which brain regions are most altered in their size in these X and Y dosage variation disorders, and do those regions make sense when we think about the pattern of behavioral change we see? The problem we immediately hit is that sex chromosome aneuploidy has a walking effect on your total brain size. So there's almost a 20% difference in total brain volume between the largest and the smallest groups here. And that's a problem because every single brain region in XY disorder is on average smaller than XY. So we needed to address this question of how to resolve changes in regional brain anatomy while dealing with total brain volume. Now, the classic approach for doing this is proportionalization. So think of the big gray square as the total brain and the red square as the bit you're interested in looking at. The proportionalization approach would just divide the size of the red by the size of the gray. And this is the average configuration in XY and XXY in this cartoon. So if you approach this question using proportionalization, you would conclude that adding an X to a male increases the size of the red region. The problem is, though, that that assumes that the correct null, i.e. what would the brain look like in an XY individual who has the same brain size as an XXY individual, is A. So the assumption here is that the proportions of the brain are invariant across different brain sizes. And the question is, well, is that a safe assumption to make? Are brain proportions constant across different brain sizes? Well, if we look at comparative literature, the answer is a resounding no. So what I'm showing you here is mammalian species ordered on the x-axis by brain size, and I'm showing you the fraction of different telencephalic regions on the y. And you can see there's a systematic shift in brain proportions as a function of brain size across mammalian species. This phenomenon is also seen within primates. This is a beautiful study by Hill and Van Essen, which asks how does cortical shape get reorganized by brain size variation through primate evolution and through primate development. So in the top, they're contrasting regional cortical surface area in adult humans and adult macaques, and in the bottom, they're contrasting regional surface area between an adult human and an infant human. And you can see that both of these major axes of brain size variation in primates induce this same reorganization in cortical surface area, where you get a disproportionate expansion of these yellow regions at the cost of relative surface contraction in the blue regions. So coming back to the clinical question that motivated this, we needed to know, well, actually, how does this work within humans? What happens to brain proportion as a function of the almost twofold variation in brain size you see in typically developing humans? And if we answer that, then we can better test for regional brain abnormalities in patients with altered brain sizes. So the problem was there wasn't really an answer to this question at the time that we needed it. So we decided to have a look. And this paper involved a massive collaboration with a large number of individuals and labs. It was really great fun to do, and it was led by two amazing PhD students in the group, Jacob Seidlitz and Kirk Reardon. And what we wanted to do is say, how does human brain shape vary as a function of human brain size in health? So the starting point was to take 3 large independent structural imaging data sets, the Philadelphia Neurodevelopmental Cohort, 1,300 cross-sectional 3T scans from youth, the National Institutes of Health longitudinal sample, 1,500 scans from 800 people, 1.5T scanner, and the HCP, just over 1,000 scans in 3T. And in these analyses, we took age and sex effects out of the data. So we took these 3 data sets and we submitted them to standard pipelines for surface-based morphometry, FreeSurfer and Civet. The results didn't vary depending on which pipeline you used. And this is the starting point for our analysis. We use these pipelines to measure surface area at tens of thousands of points on the cortical sheet in each brain, as well as the total cortical surface area of each individual. And then we were set to answer our question, how regional brain anatomy varies as a function of total brain size. And we did this in a log-log regression framework. So let me just walk you through this. The way you do it is you look at the log of the vertex area and you relate it to the log of the total area. If that slope is 1, it means that the size of the vertex proportionally is stable across brain size. If you double brain size, you double that vertex area. If the slope is greater than 1, it means that vertex becomes disproportionately expanded in larger brains. And if the slope is less than 1, it means the vertex becomes relatively contracted in larger brains. So what we wanted to do is to make and look at a map across the cortical sheet of this scaling coefficient, where red means that the region is disproportionately expanding, blue means it's relatively contracting, and white means it's scaling linearly with brain size. So what did we find? Well, we found that there was this striking, regionally specific patterning of cortical scaling. I'm showing you the results in the Philadelphia Neurodevelopmental Cohort here. And where you see red in these dorsolateral prefrontal, angular gyrus, lateral temporal, precuneus, and anterior cingulate regions, these are disproportionately expanded in larger brain humans, and the blue regions, predominantly primary sensory motor and ventral temporal regions, become relatively contracted in size. This map was highly reproducible, between data sets. I'm showing you the conjunction of the map between the PNC and the NIH cohorts, and they were highly aligned using the spin test. So this network of blue regions becomes disproportionately small, and the network of red regions becomes large. So the question was, well, what does this map mean, this highly reproducible, stereotyped reorganization of cortical surface area? So to try and get at this question, we use the spin test to compare the spatial patterning of this map to the spatial patterning of other fundamental properties of the cortical sheet. And immediately, the first thing we did is go to that Hill and Van Essen map, which I'm showing you projected onto a different cortical surface here. And the profound thing for us was when we saw that the way cortical area gets reorganized as a function of brain size variation in primates closely recapitulates the way cortical surface area gets reorganized along the two other axes of primate brain size variation, evolving a bigger one or growing a bigger one. What this suggests is there's a fundamental blueprint within the primate brain by which, if you have a bigger one, you tend to need to reorganize your cortical surface area in this way. So what is it about this map of cortical surface area reorganization? Well, we can combine this with the Yo parcellation and found that the hyperexpanding regions tend to sit within the default mode network, which we've heard about in Danny Bassett's key talk, which is this distributed functional connected network within the brain that plays a sort of important role integrating information across lower order brain networks and kind of orchestrating lower order network dynamics. This theme of integration was reinforced when we compared the scaling map with a cytoarchitectonic map projected to the cortex. So these expanding regions tended to lie within von Economo cortex type 3, which is notable for having this expanded supragranular thickness, which is thought to be important for long-range cortico-cortical connectivity. So hyperexpanding regions have this information integration signature. They also have a cost signature. They soak up a disproportionate amount of glucose and arterial blood flow at rest. And these themes of biological cost and importance for information integration were reinforced when we compared the spatial map to maps of gene expression in the Allen Atlas. The hyperexpanding red regions had unusually high expression of genes that localized the synapses and the cell's energy plants, the mitochondria. So to summarize this first section, prompts from a clinical question led to new basic science insights regarding anatomical scaling of the human brain, which prompt a whole host of subsequent basic science questions. But I want to focus on two implications of this map for clinical science. And one, they provide new theoretical context. So as many of you will know, there's this emerging finding that multiple mental disorders seem to be associated with structural and functional abnormality within the default mode network. So what our data show is that this potential network of convergent vulnerability in psychopathology is really the same network that's disproportionately expanded as part of increasing primate brain size. But coming back to where we began, this information now lets us properly go and try and pinpoint where these genetic abnormalities impact regional brain anatomy, which leads us on to Act 2, in which these normative models yield sharper maps of disease. So what we've been doing is I'm not going to show you here, but we've been systematically mapping these scaling coefficients in health across the whole brain, across different components of the brain, and then using this to help us pinpoint shifts in proportional brain anatomy in these genetic disorders that we study. So this is a series of studies led by Catherine Mankiw, Ajana Dig, Kirk Reardon, and Anastasia Xenofontos, amazing students within the lab. And what they showed us is that if you look at patients with increasing X and Y chromosome dosage, they tend to have a disproportionately reduced size of the cerebellum, the pallidum, and the amygdala, whereas the relative volume of other brain regions doesn't seem to care about sex chromosome dosage. But if you left with that message, that wouldn't be accurate, because you can then ask, what about the organization of structures that aren't changed in their total size? If we control for the size of the cortex, is there a reconfiguration of cortical shape? And I'm showing you that in these obligatory spinning brain maps here. So you've got the cortex at the top and the striatum at the thalamus at the bottom. Now, where you see white, the proportional size of these brain regions couldn't care less how many X and Y chromosomes you have. Where you see a color, these regions are sensitive to the dosage of sex chromosomes, and the color encodes the nature of their sensitivity. So these red regions change in proportional surface area as you add an X or a Y, The green regions shift just when you add an X, and the blue regions shift just when you add a Y. So just to step back here and put this in context, in that Hadron Collider experiment, I was saying we can look at different genetic disorders and see their patterned effects on phenotypes, and thereby try and retrace this breadcrumb trail from gene dosage to neurodevelopmental disorder. Well, what I showed you at the beginning is that this patterned effect of X and Y chromosome dosage on different domains of cognition and behavior, particularly striking language and social function. And that's accompanied by this spatially patterned effect of X and Y chromosome dosage on regional brain organization. So the question is, are these brain regions in any way preferentially related to cognitive domains that are relevant for the problems we see in the patients? And the first crack we took at this a while ago was using the amazing Neurosynth platform developed by Tal Yarkoni and others, which lets you do many wondrous things, but one of them is that you can submit a set of brain regions or a brain map, and you get that sort of decoded. You get back a list of cognitive terms that tend to be associated with those brain regions in functional imaging literature. So just to orient you, these rows are the brain regions that show convergent vulnerability to sex chromosome aneuploidy, and these columns are cognitive terms associated with one or more of those regions. And the color encodes the strength of the association. So when you cluster this matrix, what it tells you is that map I showed you, those brain regions that seem to be particularly vulnerable to sex chromosome dosage are preferentially involved in detection and processing of biological motion, language, autobiographical memory, and reward affect and interoception. And this is important for us because these sorts of cognitive domains, at least in terms of face validity, seem highly relevant for that targeted effect of aneuploidy on language and social functioning that I was mentioning. So just to summarize where we've got to so far before the final piece, we began trying to resolve regional brain changes in these genetic disorders as models for the psychiatric conditions we all care about, or many of us do. We came across this problem of brain size. We wanted to solve that by understanding the relationship between brain size and regional anatomy, which unveiled these scaling laws within the human brain. These laws then helped us to pinpoint which brain regions are most sensitive to X and Y chromosome dosage. And these regions seem to be relevant for the sort of behaviors that are impacted most in patients. The question that we were left with then is, but what determines the regional vulnerability of the cortical sheet to a given neurodevelopmental disorder? So what really blew our minds is that you can add these large gene dosage abnormalities, really impact cognition and behavior, but there are bits of the brain, these white bits in the right map, that really couldn't care less. Why is it that it's those colored regions that get altered and not the white ones? And if you can understand that, that gives you a real insight into disease mechanism. If you can understand what makes one cortical region more vulnerable than the other, and potentially identify molecular pathways that you can manipulate to ameliorate the impact of the neurodevelopmental disorder. So that's the question that was kind of buzzing in our minds. And that's what we're going to address in the final data part of the talk, in which spatial analysis decodes regional brain vulnerability to neurodevelopmental disorders. So, again, the organizing question here is what determines the spatial patterning of anatomical change given a certain genetic disorder? And when we looked around the literature, there were some real clues that the transcriptional vulnerability model might explain the spatial targeting. And let me just talk you through these studies. These are studies in idiopathic psychiatric disorders, not genetically defined, led by Rafa Romero-Garcia and Richard Bethlehem, who's here, and Sarah Morgan and Ed Bullmore, using methods developed by Jacob Seidlitz that I'll come on to in a moment. So what these studies did is they began by mapping anatomical change in a psychiatric disorder. So Rafa's Romero-Garcia's project mapped cortical thickness change in autism, and Sarah Morgan's project mapped morphometric similarity change in schizophrenia, and I'll come back to morphometric similarity. The next step was to take these maps and then align them with the Allen Institute adult human microarray dataset, and to find genes that have spatial expression patterns in health that are most correlated with the pattern of anatomical change in the disease state. And when they looked at those genes in each condition, what emerged is that those genes that in health are expressed in a pattern that looks like cortical change in autism are enriched for genes that are actually downregulated in the brains of individuals with autism in post-mortem work. And similarly, those genes that are expressed in health in a pattern that looks like the morphometric similarity change in autism are enriched for genes that are known to harbor risk variants for schizophrenia. So what this means is that the intrinsic spatial patterns of gene expression in the human brain can constrain the spatial patterning of disease effects. Now, genetically defined neurodevelopmental disorders like sex chromosome aneuploidies uniquely allow tests of the transcriptional vulnerability hypothesis in humans because the ground truth is known. So it's essentially a quasi-experiment. Nature has altered gene dosage in certain individuals. You know which genes are altered. You can map the altered brain anatomy in those patients and say, well, is that map of altered brain anatomy best predicted by the genes that I know have caused the disorder, that have been altered in the condition? And that's what we wanted to pursue. And we did that using this wonderful method developed by Jacob Seidlitz, a fabulous PhD student in the group, called morphometric similarity network mapping. And Jacob's insight, he wanted to try and find a way of developing structural networks in individual brains from structural MRI scans. And his insight was, well, we can develop a network within a single person's brain by calculating the similarity between any two regions across a set of morphometric features. So in one brain, in every region, you can measure cortical thickness, surface area, sulcal depth, curvature, for example, and then use the correlation in these features to quantify how morphometrically similar any pair of two regions are in that person's brain. And that yields this individual morphometric similarity matrix. If you average the rows of this matrix, you get a map in one person's brain of mean morphometric similarity of each region. The red regions have high average morphometric similarity to the rest of the brain. The blue regions have low. And then you can average this to the group level too. Now, this was a very attractive phenotype for testing the transcriptional vulnerability model because it gives you person-level maps and a group average map. It's robust and reproducible, and Jacob showed that gradients of morphometric similarity closely recover gradients of cytoarchitectonic types in the cortex. And the edge strength in this matrix here is related to the strength of gene co-expression between two regions and the strength of axonal connectivity in primate track tracing data. And that inter-individual variation in the pattern of these maps can predict inter-individual variation in IQ. So many features that suggest this in vivo anatomical phenotype is getting at some microstructural features of the cortex and is behaviorally relevant. So we sought to use morphometric similarity as a method to test this transcriptional vulnerability model using our genetically defined disorders. And I want to emphasize, this all probably sounds super niche. I'm just realizing how niche it sounds as I'm saying it now. But actually, what we're getting at is that general principles of cortical vulnerability. We're using these naturally occurring experiments to try and understand what determines which brain region gets hit by a particular insult, developmental insult. So this work is on bioRxiv. If there are any reviews in the audience, I hope to enthuse you even more. So the approach here is that Jacob began with this phenomenal data set of multiple genetic disorders with matched controls. And he calculated morphometric similarity networks on these individuals, and the output was 6 maps. So each of these maps shows you what happens to morphometric similarity when you change a different set of genes. So I'm showing you the morphometric similarity change map when you add an X, when you take away an X, when you add a Y, when you add 21, Down syndrome, or when you take away 2 small chunks of chromosomes. So velocardiofacial syndrome is missing a bit of chromosome 22, and Wagner syndrome is missing a bit of chromosome 11. So we know the gene sets that cause these anatomical change maps. The next step was to align these maps to the Allen Atlas. In our case, 15,000 maps of expression for 15,000 genes. And then for each disorder, you align the anatomical change map to the Allen map. And we used partial least squares regression to rank the 15,000 genes in the Allen by their spatial correspondence to the disease anatomical change map. So let me just orient you to this ranking because it's important for the next slides. Genes in the middle of the ranking have expression patterns in health that bear no spatial similarity to the disease map. Genes at the extremes of the rank have spatial patterns in health that are correlated to the disease map. The direction of that correlation differs depending which end of the rank you are. So genes with a high positive rank, where they are high expressed in the brain, patients show reduced morphometric similarity, and the opposite is true at the other end. So our question using this rank list is, are the cortical anatomy changes in each of the 6 CNVs preferentially coupled to spatial expression of genes in those CNV regions in the healthy brain? And the quantitative test for that is the median rank of the CNV gene set extreme relative to null expectations? Well, to our amazement, the answer was yes for all 6 disorders we studied. I'm only showing you for the X chromosome just for time here. So this is the morphometric similarity change map when you add an X to a human. And this is the median rank of the genes in each chromosome in terms of their spatial correlation with that map. And conspicuously, the X chromosome had the most extreme rank. And this same principle was found for each of the 6 other conditions we studied. So the model that this supports is that intrinsic gradients of gene expression in the human brain shape regional cortical vulnerability when you alter gene dosage. So in health, we've all got 2 copies of most genes, and each of those genes has a map of spatial expression on the cortex. Now, if you have a person that you add some genes to, some of those spatial maps then become relevant in driving the regional anatomical change and determining the targeting of the disease on cortical anatomy. So this was a really sort of stirring insight for us, but in a way, it only just pushed the question back one step further, because the question becomes, what's determining this? What principle of cortical organization is organizing these gene expression gradients? And the strong clue from the literature was about cell type. So what we already know is if you take a chunk of cortex and measure its bulk gene expression, and you take another chunk of cortex and measure its bulk gene expression, and you compare them, the dominant reason for the difference is which cells, what the cellular composition of those chunks of cortex was. So what we wanted to try to do is to deconvolve cortical expression with respect to single cells. And the hope here is, could we then potentially associate each disease map with a cell or a set of cells? And that would be a major advance if we could try to, at least. So Jacob did just that, undertook this Herculean effort of trawling all of the large single-cell gene expression studies of adult human cortex and gathering 50 cell-type gene sets. So there are 50 cells, and each one, the people publishing the study said these genes tag this cell type. And for each cell type, we then use the Allen Atlas to calculate a map of expression, so 50 expression maps. And when you cluster those maps by their spatial correspondence, you get back all 7 canonical cell types in the human central nervous system. So Jacob used this approach to make a map of astrocyte expression, of endothelial cell gene expression, microglia, excitatory neurons, inhibitory neurons, oligodendrocytes, and oligodendrocyte precursors. And now we were ready. When you have this map, you can then say, for each disease map, which cell type map looks most like this disease map. And this is the final data result I want to show you, where we did just that, and this is the result. So just to orient you, these are the distinct genetic disorders, and these are the cell types. Where you see a dot with a gene, it means that that cell type map is unusually correlated to that disease map. And we placed a second requirement. For each disease, we said, tell us which cells have expression maps that look like the disease map. And then from those cells, only give us the ones that actually express genes from the CNV. And that's the genes that you're seeing here. So what this is doing is it's using readily available patient structural neuroimaging data, publicly available atlases of gene expression and single cell expression, to try and make cell and gene level specific inferences about what's driving regional anatomical change in these disorders. So to make it concrete, our data suggests that if you're going to look anywhere, with respect to the cortical anatomy change in trisomy 21, the blue regions show high expression of oligodendrocytes, which contain the chromosome 21 gene PCP4. So it's nominating a cell and gene set specific driver of the anatomical change, which obviously needs validation in post-mortem data from patients using single cell, which we're starting to do now. So just to wrap up, in this section of the talk, in this final section, I think it kind of samples all arms of the strategy that I've been advocating. So we use large-scale studies of brain development and health to develop this novel metric, morphometric similarity mapping, which was tuned to the features we were interested in, gene expression and cellular features. Took that method and used it to map the effects of disease, different genetically defined neurodevelopmental disorders on the human cortex, and then merged those maps with maps of cell expression and gene expression to make inferences regarding molecular and cellular disease pathways from in vivo structural MRI. I hope in the course of this talk I've enthused you about the general principles that I've described. Again, to emphasize, I was showing you their specific instantiation with regards to the disorders we study, but I hope and believe that these methods seem relevant to many of you as you think about the conditions that you study. Of course, there's much work to be done. With regards to the imaging phenotypes, we want to now extend beyond the humble but beautiful T1-weighted structural MRI scan and try to use multimodal imaging data a bit more wisely, applying multivariate methods, and critically, developing anatomical phenotypes in health that are tuned to the components of brain biology that we think are going to be relevant for disease, and then take those phenotypes into patients. With regard to the Hadron Collider approach, I think we simply just need more genetically distinct groups with the same data and to cross levels to understand how dysregulated gene expression in patients can map onto dysregulated cognition, for example, and to bridge from these genetically defined disorders to idiopathic conditions. And then finally, with regards to the spatial integration, I think the task facing us, and it's clearly going to move at a massive pace now, is to generate and combine in a very data-driven way, multiple maps of microstructural, molecular, chemoreceptor, gene expression maps of the cortical sheet. Take all of these microstructural data, project them into standard imaging space, and use that as almost standard practice to sort of decode some of these maps that we're getting. And ultimately, with the hypotheses that we form that way, after having cast a broad net, then be able to engage in very expensive work to actually drive in and test, validate in post-mortem, in rare post-mortem tissue from humans. Are we right? Is it that gene in that cell in that region? So with that, I want to acknowledge the amazing team of people that did this work and the collaborators that we're fortunate enough to work with. To highlight posters that the team are going to be doing, please come by and say hello, and to make a shameless plug for 2 postdoc positions, and to also thank Shirley and Alma, who I miss very much, and without whom none of this would be possible. Thank you.
I think we're right at the limit of time for this session, so I don't think we'll take any questions, but Armin has agreed to stick around a bit if anyone wants to come up and ask him something personally. Thanks again, Armin, for a great talk, and enjoy the rest of your day.